Well, welcome all of you. We're here on the traditional land of the Huron-Wendat, the Iroquois, Haudenosaunee, the Anishinaabe, and the Mississaugas of the New Credit. And, you know, the land is storied. The land has its own secrets. And I think that, you know, it's always important for us to acknowledge who's been here before, and also to recognize the fact that they're still here. Like, we're all here. Takaronto is one of the names that this region was known by, and it means the place where the trees gather in the water, right? And it's a gathering place, and so we're very grateful and to be able to live and work here. And welcome all of you to this place if you haven't been here before. I'd like to introduce Radha and give her a chance to talk. You know, we're recording this for archival purposes, so I just want you to know that. But you're not being filmed, so don't worry about that. We will have a Q&A at the end, so you'll have a chance to ask questions. But I'd like to just ask people to put their phones on silent so we don't have interruptions during that. And then I'd like to introduce Radha Chaddah, who is a visual artist, a multimedia artist, a scientist from Ontario, born in Owen Sound, and you know, works in both the fields of science and the arts. And I think this is a very exciting time for us to be looking at practices that do that, because it's time we broke down some of the Cartesian separations between disciplines and find some solutions to the big challenges we all face. And I think artists like you have some of the answers we need to be looking at, or, you know, not maybe the answers, but certainly help us transform the way we see the world. So thank you. Welcome, Radha.
Thank you. Thank you very much for that kind introduction. Thank you to the Aga Khan Museum for inviting me, and to Amirali Alibhai for inviting me here, and also just you know, being with us through this whole collaborative work, which is YAM. So Amir asked me to tell you a little bit about my practice before I make an introduction to the films that you're going to see today, so that's what I'm going to do now. So my path to now began in the arts, where I studied film and art history at Queen's. And this is where I really became compelled by the power of the visual image. And I took a hard detour through research science, where I studied cell and molecular biology, about stem cells really, and at a neurobiology lab at U of T. And so after I made that bridge from science back to art, I took my first ever exhibition to the Isabel Bader Gallery at Queen's University. And this was one of my first exhibitions of my artistic work. So this is something that happened while I was at the University of Toronto. I fell in love with immunohistochemistry. So these cells that you see here are called astrocytes, and it's a central nervous system cell. So these cells sort of, they're master regulators. They reach out and grab onto bodies of the axons of neurons, and they reach out and they grab onto blood vessels, and they're kind of like a communicative unit, and they sort of clean up your metabolic waste on a daily basis, et cetera. Fascinating cells. And the thing that I like the most about peering into this kind of world of invisible materiality were the surprising truths of this reality. So you think of cells as being these little fluid-filled sacs, but in fact, what you see in yellow here are the bones of the cells, or the skeleton of the cells, decorated with small fluorescent molecules. So this process of staining cells, obviously, then needs to be visualized. And at that stage in the laboratory then, at this stage in my work, I was using scanning laser confocal microscopes to visualize these images. So here you can see this system, and there's a kind of a pulse of laser light that hits the samples, and then the machine will scan pixel by pixel, line by line across your sample to produce a single kind of high resolution image. So these images were tiny and designed for print in scientific journals at sort of 1 inch by 1 inch. And I became just fascinated with cell photography and the expense of data collection. And I spent quite a while in the lab, and what I really wanted to do at the end, to bridge my way out of science and back into art, I sort of recreated this technique of immunohistochemistry in the laboratory so that I could purpose it as a kind of a novel artistic medium. And what I really wanted to do was reverse the scale of this microscopic realm and produce large objects and photographs that people could kind of stand in front of and see, engage with their own material reality in front of. So this was my first show at and the reason I'm showing you this is because, you can see how these photographs are printed, but this image is actually a stack of 7 singular photographs all smashed together. So what you get is this depth, and what you're looking at is hundreds of thousands of cells that are growing out from neural stem cell colonies in culture. So it wasn't long before I developed a full-blown love affair with the University of Toronto, and there's a medical imaging laboratory, and I broke out of the mold of fluorescence microscopy and started to explore some of the other machines that they had there. So what you just saw there was the inside, the guts of an electron microscope. And the fluorescence microscopy that I was previously using, a very colorful fluorescence microscopy, is sort of limited in scale to a power of about 600 times. And I was compelled by that, by these scanning electron microscopes, because they can see so deeply. And there's a new system at the university that was purchased that could see 1 million times. So the sample preparation technique is quite different for this microscope. You take the samples, and they need to be dehydrated, and then they need to be actually coated in gold. So you dehydrate the samples, you put them on a tissue block, and here you can see there's a little block, and you can see each one of these little individual samples. Some of them are seeds. There's a piece of rock. There's the underside, or a leaf specimen. So you prepare the samples like this, you stick them down, and then this is one of my all-time favorite machines at the university. It's called a sputter coater, and you put the dehydrated samples into this sputter coater, and in the middle you can see sort of a cylindrical chamber, and that's where the samples go into. And at the top of the chamber, there's a gold bar. And what you do is you suck all of the air out of the chamber, you pump it full of a gas, argon gas, you run a current through the machine, and the gas becomes ionized or charged, so it splits into positive and negative particles. And there's a gold bar at the inside of the chamber with a magnet behind it, and the magnet effectively sucks the ions up toward it, and it interacts with the gold bar at the top of the chamber. And so it peels off tiny ions of gold that sprinkle all over the samples that are rotating in the chamber, and you get a really, really fine layer of molecular gold coating the samples. So I'll show you that. So essentially what's happening is it's this plasma inside this chamber. There's no electricity, there's no light, there's no, well, other than the current that you're running through, but it's not lit up with a light. It's actually the glow of the plasma, which of course is what northern lights are, charged gas. So once the samples are removed from this container, you can see they're coated in a tiny and a very fine layer of molecular gold. So then you can take it into the microscope, And of course, a scanning electron microscope is not a light-based microscope. It works by bombarding the sample with a beam of electrons. The electrons scatter off the surface of the sample, and it's that pattern that creates an image. So this is not light-based photography. So here you can see, this is actually a speck of wax that is about the size of a sesame seed that I took from my husband's beehives. So there's a kind of a little drawer at the bottom, and it's sort of like a bee toilet. And I was interested to see pollen using the scanning electron microscope because it's beautiful, and there are many different varieties of shapes. And so I went fishing for pollen, and this is what I found. So the nice thing about this machine is that you mount the samples on a stage, but the stage isn't flat like the other light-based microscopic systems that either hit your sample from above or below. So it bombards from this direction, but you can turn the stage in a U-centric way from a central point. And in this way, you can tilt the stage, and then you can go in photographically deeper, 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 and there's kind of a greater sense of depth because of your ability to tilt the stage. So when I went in about 1,000 times, this is what I found. This sort of compelling world. And at first I didn't understand what I was seeing, and it took me some time to sort of research and be able to identify the structures that I saw. And you can see some little round structures, those are grains of pollen, and you can see these little kind of pearls on a string, which are fungal spores, and there are little broken down bee parts, etc. And this was part of a show that I made it into a show called Beneath Us. And while I was shooting some of these photographs, I was working on a play and collaborating with a composer called Dan Bedard. And he started playing with some of these photographs, creating music from them. And so he would use a contrast strategy to create a topographical kind of map out of the photograph. And then he would push sounds through them. And he would come up with these kind of resonances that he used to compose music. So I'm telling you this because some of this music was stitched into the pieces that you're going to see tonight. Some of this music was created the same way. So this show, Beneath Us, was really about our place in nature. And it's about how, from my perspective, I think that, our perception about our place in nature is upside down. Everywhere I look in terms of human society, the way we organize ourselves, I see pyramids everywhere. But really, it's the bees who we think are beneath us. All of these other creatures we sort of think are beneath us. At least we act that way if we don't think that way. It's the bees that, of course, support us and our very existence. And so this show was really about taking steps toward reframing our human relationship with nature and sort of recontextualizing our place in nature, or re-examining that. So back to the scanning electron microscope and a whole other body of samples that I collected on my travels, this time to Maui. And this is the underside of a wauke leaf. It's a very important plant culturally and medicinally. It's used to make a traditional cloth called kapa. And this is the underside of the leaf, and you can see all these little hairs that are used as defense against insects. And I'm showing you this because this underlies my process, kind of going out into the world with my own curiosity, collecting various bits and pieces, taking them into the laboratory, imaging them, and then typically what I like to do is reverse the scale of these things. So here is a picture of the underside of that wauke leaf projected in my outdoor studio onto grass with a massive stand of 200-year-old sort of cedars behind them. So back to the scanning electron microscopy. I wanted to show you this one because it leads into my presentation of EM. So this is a stamen from a how plant. And so you can see at the top end there, these kind of like little Velcro pads. And at the bottom end, this is the root that I pulled out of the flower. And if we take a little trip in, you can start to see these little grains, these little sort of spherical, spiky grains. And along the bottom, there's a bar with many of these images. Sorry, I should have told you that before. And the bar will show you the scale. So you can see there that's a millimeter, that little length. And over here, you can see the power of magnification 52 times. So if you keep going in for a little trip, I'm going to skip these next couple of slides, if you forgive me. If you keep going in now, we can see we're at about 250 times. And you can see the kind of this sticky Velcro sort of nature of the relationship between the pads and the stamen of the plant and the pollen grains that are stuck to it. And this started to interest me because, of course, I looked at these structures and I thought, it looks very much like the coronavirus. Does it look that way to you? And, this is 1,500 times magnified, but you can see the scale bar shows you the size of this piece of pollen, about 40 micrometers. So it's many magnitudes larger than that coronavirus. But what I see is how patterns repeat themselves in nature from the micro to the macro. And also of note, all of those little round dots that you see studding the surface of that grain of pollen, those are all bacterial colonies. So, and of course, as you know, viruses are many, many times smaller than bacteria. And so there will be thousands or hundreds of thousands of individual bacterium in each one of those little round colonies. So this material was all collected from Hawaii, from Maui, the samples that I just showed you. And I had a big show lined up there in 2020. And of course, it was canceled by the pandemic. So I sat in my studio and sort of looked out the window and wondered about the shape of this thing, of this virus, and wondered about the mechanisms of how it gets into the cell and how it replicates itself. And so I started reading, I put my science hat back on, and I started reading publications, papers, talking to physicians and researchers, and trying to make some sense of this very mysterious pathogen. So out of this, really, musing came a new show. And it's called Iam, which means now. And Iam is a piece about perspective in our pandemic time. And it follows the COVID story through 4 increasingly larger material realms. So we begin in the molecular, then we go into the human realm with a body and mind, and these are the first 2 acts of the piece that you'll see tonight. And then we phase into the planetary realm, and then finally the cosmic. So in the first act that you'll see tonight, it's really about the human viral dance that unfolds in our sort of multi-million year running tale of enmeshment. So amidst sort of rotating 3D molecular projections, the life cycle of the coronavirus transpires to reveal the virus's reliance on human molecules to bring it into the cell, to replicate it. It's kind of a collaborative relationship. It's hard for us to engage with that reality because our experience has been so painful, but that's the reality of it. So I think I was reading the other day that our genome is packed full of ancient viral code. Up to about 8% of our genome is actually viral code. And if not for that viral code, our mammalian bodies would not be able to do things like create the placenta that attaches into the wall of the uterus and nourishes the baby during development. So it's actually viral molecules that allow us to create that interface. So in Of Body and Mind, the second act, it's goes into the human realm, and it tells the sort of angsty tale of our human experience with with COVID. So the sort of stress of isolation and lockdown, what happened to people when they were separated from one another. Of course, this is a very fresh experience, so I don't need to fill you in too much about that act. And then the third act is called Planetary, and it's a happy story in that act, really, because the air was cleaner, the water was cleaner, animals came out to inhabit the space that we had previously taken up while we retreated to our caves. And in the final act, Cosmic, the story of COVID doesn't register. It's inconsequential. And so this is kind of this 4-act story of perspective in our pandemic time. So back to the first act, Dance of the Molecules. So as I told you, I began with experimenting, reading around and experimenting with molecular models that I found at this unbelievable repository of free medical information, molecular information called the PDSB. It's a protein database. And so I was able to use these models and play with them. So I could move them in various ways. I could rotate them around a 3D axis, as you see here. I could zoom into them. And what you're seeing here is a complex of the ACE2 molecule, which is on the surface of human cells, and the spike protein of the coronavirus, which make a complex. And that's how the virus is brought into human cells. So experimenting outdoors with this, there were these really dynamic moments that we could create when we ventured inside the molecules, and this incredible play of light scraping across the trees and the grass, which was infinitely compelling. So here you are inside a molecule, and this is a point model. So the last model you saw was a bonding model, which shows you how each of these atoms are bonded. And here is a point model showing you each atom in this molecule as a singular point of light. And now zooming out from that, we can see this is a single spike on the surface of the coronavirus. This is what that molecule is shaped like. And of course, this is the code or the recipe to make this molecule is what is encoded in the vaccine that we've all had. So we began playing around with these, I began playing around with these 3D projected images in my outdoor studio. And at a certain point, sitting around the dinner table with my family, my one daughter, as I talked about it, and I talked about this viral human molecular kind of interplay and the dance, and she said, "Mama, I think it's a dance." So at the time, I was working with Jacqueline Bloomis, who's a musician and my intrepid co-producer on these pieces, and she brought in her sister, Ali Bloomis, who works with Open Fortress, and is a fantastic choreographer and dancer, and she came on board to start in the experimental stages of the process. And it was a beautiful moment because we kind of made under 5 studio, and we were in the middle of the pandemic, and it was early times, and people were so afraid, and all we wanted to do was to continue to make art. And as a small group of creatives, we were able to adapt. And we decided, okay, we'll work outside, and we'll work in groups of 5 or less, and we can keep our masks on, even if we're outside or inside. And in this way, we can make our way forward in a comfortable way that can't be shut down. And that's what we did. We just kept going through all of the openings and closings and everything. We just kept going. So here's Ali. And I think as soon as I started putting these projections on the trees, I knew that, I could see how beautiful it was and how compelling it was. But this was the moment when Ali started to move, and we spent a lot of time talking about molecular machines and their movements, and this informed Ali's choreography. But here, this moment, when she started to turn with this turning, rotating 3D molecule, and something so magical happened, I knew that it was going to be good. Here's another moment in the beginning. We experimented a lot with speed and the speed of the dance and the pace of the projection. So this is obviously a little, well, it's not obvious actually, but it's a little slow-mo video of Ali in the very beginning stages. Sort of imagining if she were a molecular machine or what she would move like. Then we brought in the intrepid ladies from Call and Response and Call and Response Clothing, and they came in to work with us and collaborate on the costume design. And this was the very first sketch of what the coronavirus could look like. And so they were challenged by what the coronavirus is. So it's basically that spiky shaped molecule that you saw that's a similar shape to that piece of pollen, but that's kind of a coat on the outside. And the coat gets peeled off as after the virus enters the cell. And what happens, the coat gets peeled off and the viral code, that little short strand of mRNA, it gets inserted into the cell, and the virus kind of floats naked as just a piece of code when its coat gets stripped off. And then human molecules wrap around that little viral piece of code and they replicate it. And then the virus, they replicate all different parts of the virus. So they replicate the code and they replicate the coat, and then the virus is packaged up again in its coat, and it's exported from the cell. So here you can see here, their concept for the costume clearly included a coat. And in the film, at one moment, you'll see how the ACE2 receptor, that character, they kind of get stuck, and the ACE2 receptor peels the coat off the virus. And that really is a metaphor for how it happens in the cell. And after that moment, then she sort of floats free in the cell. Here's just a little moment where we're fixing Ali's, you can see how the costume sort of came to life, and fixing the collar on Ali, the coronavirus character, her costume. So color was a very important consideration for us. Always bent my mind a little bit to compose on a screen and use color that way, and then you project onto trees, which of course are green, and everything is upside down. So what looks great on the screen doesn't look great projected on trees and grass. So it's always a back and forth of experimentation to figure out what works best. For this piece, for IAM, what we did was we created an immersive light space out of projected light onto trees and grass, and the dancers play in this space. So the stage is about 50 feet high and 50 feet deep, and it's about 80 feet wide. And so you can see here we're just setting up. We've finished the creative development, worked through the summer season after the leaves emerged to the very end, and then in the fall of the first year of our production, we filmed the piece because we were still all locked down and there was no way for us to get this work to the public. And we felt strongly about having something to offer. And so we chose to film it again with, small team of people, and produce it as a film so that we could bring it to people. So here you can see what the stage looks like without projection. And here you can see, this is actually an image from the second act of Body and Mind. It's a tissue shot. The large round structure you see in the middle is sliced through a long bone with marrow inside, and then all around is the muscle or the meat. Here's our director of photography, Henry Sansom, setting up for the shoot. And they were very long nights, of course, because we couldn't start filming until after dark. And so we kind of just pushed through until the wee hours of the morning. So here's a funny shot of Henry And, you know, this is 5 in the morning on our last night of filming, and we just decided to play. And some of this footage made it into the film, and Henry has quite a playful spirit, and for me, that's a really important quality in a collaborator. So, on to the second act, the introduction to the second act. So, as I told you, this act is the human story of kind of our suffering and isolation that we experienced in lockdown. And this story actually strangely began with a different body of work that I was working on, but it's also a tale of suffering. So it occurred to me through my trip through research science, I really was quite shocked by the scale of waste and also by the animal suffering that I found there, even though there are quite rigid protocols and strict protocols to protect animals in those settings. You know, what we can do to animals in a laboratory setting is shocking, and the average person has no idea. So thinking about this issue of waste, waste of animal life, mountains of garbage, I asked in the laboratory that I was working in if whoever was doing animal experiments could harvest the organs from the mice that they were using. And I wanted to make something out of these tissues from these animals that would go in the garbage. So the show was called Recycled Mouse. It was also a pandemic casualty, but little did I know that I was busy photographing for something that I hadn't imagined yet. So I included this slide. Maybe just take a little read. So, you know, it's a funny if creepy little comic, but this is, I remarked, one of the classic ways to take the sting out of what we do in science. You know, you have a little laugh. But also, for me personally, that's why I decided to really just focus all of my cell and molecular biology research using cell lines and using human cells, because I felt very strongly that the research is for us, so we should be prepared to use our own cells. So here we are, a brain in a tube. This is a mouse brain that was headed for the garbage. And I'm showing you all of this sample preparation because this is what went into creating the projected imagery for the second act. So of body and mind, while the dancers play on the stage, this kind of disconnect, this kind of a synchrony and asynchrony of the body and mind when we're sort of out of sorts or stressed. The tissue that I project, that we use to create the light stage, or the imagery, pardon me, is all tissue. So it's nerves and bone, like you saw, and meat and intestine and kidney, etc. So this is how I prepared all of these samples. So each one of these little cups is filled with a different kind of organ that's been harvested from mice. Well, harvested from the garbage, I'll say. So here is, this is called a belly shaker. And so if you are preparing samples like this, you put them on this table, and it spins the sample down to the bottom of the cup and kind of gets rid of the bubbles. So next step is to use a cryostat, which is like a very sophisticated freezer with a knife inside. So it's a frozen chamber. You open the top. Once you've shaken all the bubbles out of those little samples, you have an organ in a cup. You put them in a negative 80-degree freezer, and that colored liquid that you saw is a kind of cryomatrix that will just snap freeze. And then you can push the block out of the cup, and you can use a little bit of the cryomatrix to stick the block or onto that mount there. And then what happens is you can slice with a razor blade, and this paper-thin, much finer than paper-thin slice of what's kidney, it's kidney there, comes off onto that stage. It's very finicky work. So now you can see, once you've got that little slice onto the stage, then you kind of coax it off with a toothpick and a tiny paintbrush, and it sort of melts and adheres onto the slide. And once it's melted and adhered onto the slide, then you can start the staining process. So that staining process takes a couple of weeks, and I go back to using small fluorescent molecules for these tissues to stain them. So I image these tissues on a completely different microscopic system. And the system actually, this one is showing you the standard fluorescent system, which images pixel by pixel, line by line in one magnified field of view. But the microscope that I used actually allows you to magnify to 400 or 600 times, take one small high-resolution image at that scale, and then you can move across the entire sample. So to image that one slice of kidney that you saw, it would take about 3,000 photographs, high-resolution photographs. And then those photographs can all be stitched together to make one mega image, which could be somewhere between 2 and 7 gigabytes for a single photo. And that's what we use. We use these macro photos for the projected imagery in the second act, so that we could project at a massive scale, but we could also zoom to create movement, or zoom in to create movement, zoom out or in. And also we could go for these kind of meandering trips through the tissue. So here's a meandering trip through the tissue, just shot off the screen of, and this is a slice through that, an image of that piece of kidney that I saw you, and it's really, we're really close in that kidney, deep into that kidney, and you can see a cross-section of a renal tubule there, a collecting tubule. So here I'll play this video, and you can see what we wanted to try and sort of recreate with the projected imagery. You're starting to be able to recognize that kidney bean shape of the kidney. So this is a single image. So we were able, this single image, 7 gigabytes, let's say, and built of 3,000 different photos. And so this is where we sort of began with the imagery. So this experimentation needed to continue in all departments of our production, and this is our projection cart that we created for the second season, and made it really easy to wheel this thing in and out of the studio. And some designers, friends of mine, made this Q&A designs. It was fantastic. And here you can see us experimenting with different kinds of fabric. And of course, if you look from right to left, you can see that, you know, differences in color of the fabric made a real difference in the way that the images register in the foreground, and they play with the background projections too. So this fabric, for example, right here was kind of like a dark gold and green fabric, and the fabric on the far right, as you can see, is just a nice crisp white. So here was our sort of final costume design for the second act, which was sort of loosely inspired by a hazmat suit, and also you can see that on the left, the characters, they have this kind of pleated costume, which is sort of resembles a carapace. And on the right, on the left, here's our principal dancer, Lee Gelblum. And on the right, there's Ali Blumus, who is our choreographer as well as the dancer. And you can see Ali's costume is this sort of smooth. It's kind of references the inner life, the inner kind of emotional life of human beings, where Lee's costume references sort of the outer carapace, the outer shell, the mask. And I think I'm going to stop there with the introduction, and maybe we can screen the first act then, followed by the second act, and then we can see if you have any questions.
Thank you. That was really cool seeing it at that scale. I've only ever seen it on my phone and my computer screen. Right? And there were things I noticed, and I've seen it many times. I've seen both works many times, but I saw things I hadn't seen before. And it was really cool to hear the sound at that level and quality as well. So congratulations and thank you.
Thank you. Hope you enjoyed it.
So these journeys you talked about slightly different processes that led you through the work, and you've still got 2 more acts that you're planning to work on now. Are those being informed also by similarly different technologies? Because that's really fascinating to me, is the idea that the technology is a star, if you know what I mean, in the work itself, and that you're not hiding that technology, you're using it to tell your story.
There are different. I use different microscopic imaging systems for each of the acts. Of course, the first act uses 3D molecular models that have been extrapolated by scientists who typically figure that kind of stuff out using sort of freeze fraction microscopy, and they work out the exact shape of these molecules, and then I was able to use that free repository of 3-dimensional information, molecular information, which was fantastic. And then for the second act of Body and Mind, I told you about that Axio scanner and that kind of imaging process, creating these mega tissue images and what's behind that. For the third act, I'm going to use scanning electron microscopy of bits and pieces of nature to create the projected imagery for the planetary act. And the dancers will play wind and water. And in the final act, in keeping with what I really like to do in my work, which is exploring sort of invisible materialities, my first instinct was to take a trip into space, kind of, fly by to the edge of what the Hubble telescope can see, that kind of thing. And then I really started thinking about it, and I thought, I was more interested in representing energy that we can't see. So, of course, that all we can see is this tiny little slice of the electromagnetic spectrum, which is visible light. And even our sensory organs, our eyes, of course, are limited in that way, but they're also even limited in a dimensional kind of way, because the images that we gather are 2D. They're kind of 2D sort of images that register on our retina, and it's our other facilities that allow us to perform this kind of computational physics that lets us move through space and see objects in a 3-dimensional way. So I'll use that kind of scanning electron system for the third act, but then the fourth act, when I'm trying to represent invisible electromagnetism, of course, I need to sort of think about physics and break out of a biology lab and into the realm of sort of theoretical physics, and also working with sort of known models of the universe. So there's a group that was headed by William Percival out of the Perimeter Institute, who just produced the largest 3D map of the known universe. And so that's our starting point for the fourth act. We'll use that as a model of space and then travel within that, but not to visualize planets and body and sort of nebulas with black space, pardon me, in between, but to visualize the other electromagnetic energy that exists in the blackness of space. And the dancers will play dark matter.
I think that's one of the most fascinating things about your work is this idea of the unseen, of being able to, you know, to sort of stretch. Because when you were describing the plating, using how the electron microscope worked with, you know, bouncing electrons, you know, bombarding the sample with electrons, and they bounce off, and you create this image. It's so different from the way our biology sees things, and yet it's similar. And that, like you just mentioned, how our our brain fills in stuff or processes, you know, what is a 2D image into something 3D, or something that we know upside down and writing it the right way, and you know, like all those things that our brain does for us. But this technology is kind of like that as well. And at the very beginning, I sort of thanked you for allowing us to transform the way we see, right? And it's being done literally as well as metaphorically, because one of the things that the dancers certainly did for me was bring a very emotional, emotive, affective quality to the microscopy that you're dealing with and the technology that you're presenting, this human interaction. And I think that, you know, congratulations again. I thought that was very successful. What I'd really...
I think the congratulations actually should be given to Ali Loomis, because she created this incredible kind of language for that, for both acts. And in the second act, really, the way that she did that, she kind of, the movement, the language of movement was about struggle and overcoming. So the movements are very slow, and they're all based on breath and breath work. And so all of the poses that the dancers move through and phase through, a lot of the poses are incredibly difficult to sustain and move through. And it's only by the dancers regulating their breath and doing in this measured approach. And I mean, picture you're doing it for a film over and over and over again all night long, right? Yeah. So it was, but the whole style of movement, the choreography itself is really a metaphor for the struggle of moving through these kinds of incredibly challenging experiences.
Thank you. And I have one last question, and then perhaps we could just turn up the house lights to a little bit so we can see who's around, and we can, if anyone has questions, we can take them from the folks that are here. But my last question is really about, you know, you've created this. I had the good fortune of being able to see this work performed in the location where it was being filmed. Where it was performative, it was a live experience, right?
Well, that's what it is. It is a live performance installation, but and we're halfway through the production of it. And because we made it during a pandemic, we didn't know when we'd be able to perform it for you live. We had no idea. And so that's why at the end of the first act also we thought we want to capture this and present it, and so that's why we made the decision to shoot it as a film. And that's why we've kind of kept up, and it's kind of it's a really it's a whole other process, and you know, these are the first films I've made since I was a student, and you know, I didn't show you any of those examples because they're all super bad, but you know, it is very much a live performance installation, and we look forward to bringing you this work in 2023 and 2024. We're working on an international tour right now, and the format for the show will be all 4 acts presented together, and so that takes about an hour. And then after that, we're going to work with local musicians to come and either improvise music for you or play some of their music for visitors. And then what we'll do is we'll kind of recreate a scenography of the projected work, and visitors can walk in and experience it as a light sculptural installation, which is normally what I do. This is the first dance that I've created.
I'm very excited about seeing that when it's produced. So, if we could, because I can't see anybody out there, right? Okay, good.
Well, that's nice.
And there, now we can see people. So we have a few minutes to take any questions that might come from you. There is a microphone that Marin has over there so that we can get it recorded, your question, that is, so that if that's okay. Are there any questions, or did Radha do such an amazing job?
Are you seeing the whole picture, or are you letting it unfold? I find it really fascinating. I know you're very organized in your thought process, but I'm wondering, like, how far ahead of yourself? How much of this is also organic? Like, where do you mash up your scientific mind with your artist mind? Because it seems to me like, you know, you move through these different technologies, but the rest of us would say, wouldn't it be wonderful if I could do this? And you, Radha, always say, oh, I've got an idea. And you make it happen.
Thank you, Erin, for that question. I think, you know, sometimes I see things fully formed, not with this work, though, because there are many collaborators, and so you're always working with people and you don't know, you know, there's a bit of a mystery and stress too, but a beautiful mystery to that. But with other pieces of work, sometimes I just see the whole thing, and then it's a question of reverse engineering it, and I never know how to make what it is that I see. So, you know, I always learn new things along the way if I'm creating a piece in that way, kind of reverse engineering. But in this, for IAM, it's very much a process of regular experimentation. So we begin, you know, I began dreaming about it, reading about it, you know, and then as soon as the leaves emerged on the trees, then I started playing with these molecular models. I started with actually hemoglobin because I was interested in that molecule and its relationship to the development of severe COVID, which at that point it seemed like maybe a thing, but you know, the research sort of progressed past that. And then it's very much just a journey through playing with what you have at your disposal. So, you know, developing a familiarity with those elements, and everybody brings that familiarity and expertise of their milieu to the collaboration, and then playing and finding out what works. So, I mean, and that's a process of experimentation, and usually we do that over the entire spring, summer period, kind of, you know, I'm always experimenting, but we will all come together kind of once a week and experiment, and then sort of move forward, and then by the time the fall comes around, then we're ready-ish to film. And so I think that this is a very experimental, collaborative piece that kind of unfolds as we are making it. Yeah.
Thank you. Anyone else? There's a couple. There's one in the back and then one here in the front.
Thank you. So first of all, congratulations, and I think I'm very grateful to be here today and watching this. I think you've created really powerful work. My question is, I'm interested in learning more why you chose trees and grass as the background of your work, as the canvas of it. You mentioned how difficult it was to see the color on the screen and then projecting it and get it picked up by the camera, and it's all different. Were there any other considerations, any other options you were considering, like maybe a wall, maybe rocks? And you mentioned about the live tour you're going to be doing later on. Were you going to be doing it on other background, or it's always going to be trees and grass? Thank you.
Thank you for your compliment and for your question. So I like to project this kind of microscopic material reality or invisible realities like patterns of electromagnetism onto living structures, because for me, it's a way of communicating our enmeshment with the natural world. And so I think part of our whole struggle and the suffering that we experienced as we went through the pandemic was because somehow, you know, we have it in our minds that we can build a wall around ourselves and we can separate ourselves from nature and we can keep things out and we can keep ourselves. That's not even what we are. I mean, it's not, we are walking colonies of a lot of different kinds of organisms, and by number, there are fewer human cells in your body than there are other organisms. So what are you, you know? And so I think that, you know, we projecting onto green things and onto nature, reversing the scale, kind of this sort of enmeshment of this materiality, it really speaks to my feeling that, you know, yeah, we are not separate from nature and we cannot exist separate from it, nor can we control it. And the idea that we can is a sort of a fallacy. And I think we need to think about it a little bit more.
Thank you. And there was a question down here, Marin.
So when you tour this, are you going to be touring the film as 4 different things in gallery spaces?
No, so the work is a live performance installation, and the films are a document of what we've created. And I mean, we hope we've created successful films too, but really it's it's following along as we build to the moment where we can present this live. Yeah.
When you show the films, though?
Sure, absolutely, especially when they're all 4 done together. Then, you know, I mean, the Aga Khan Museum has been creating, obviously, digital content and storytelling around each one of the films, and we've kind of been releasing each act as it's been produced and as the films have been produced, so that, you know, we can create some build-up to the live performance. But I think next spring or in the early spring or the late winter, we'll release Acts 3 and 4 with the Aga Khan Museum digitally, so you'll be able to look at that online and see a little bit of the storytelling around it, and then the next phase is the live show.
Mm-hmm. Well, thank you. Thank you very much. Thank you, all of you.
Thank you.
I saw your hand in the back there. Did you want to?
I'm sorry I didn't tell you that. Thanks for the question. Iam means now. It's Latin for now. And so it's this kind of an arc of perspective from the micro to the macro, but of course all of these different realms are happening simultaneously. So it stacks all of these different material realms of different scales into one temporal space, which is now.
I think one of the central messages of your work really is what you said just now, and right before that when you spoke about enmeshment and that idea that separation is very arbitrary when we start to create boundaries. You know, and that observation, that fact that there are more non-human cells that comprise our everyday lives than there are human ones, and I think that's very cool. In the end, it's very cool. So, thanks everyone. Thanks for being here
Just
tonight.
blame it on the bacteria.
Yeah, that's right.
Whatever you do.
Thanks, Radha.
Thank you.